Okay, let's go ahead and get started. Couple of quick announcements. The first one is unfortunately the third candidate for the biology faculty position had to cancel, so we're not going to be having a biology seminar this week. So cool stuff about circadian rhythms we're not going to hear about. I'm actually really kind of bummed because it's a really cool story that she's going to be talking about. The second thing is, yes, the exam scantrons were scanned in. I've got all of the numbers. Have I uploaded them yet? No, I literally got them done like 3 minutes before I walked over here. But what I did do was I looked at how people did in terms of the high scores, etc. The high score was a 46, which is ridiculous that people did so well. So I haven't looked at all the rest of the statistics yet, literally because I scanned them right before I came over. And so I'll be looking at the lower scores after that. I think the next one was a 42. But even then, this exam was even more bimodal than usually my exams are. So I need to think about this a little bit before I actually talk about normalization. I will post that on D2L as soon as I have some ideas. And as I've mentioned a few times before, the final normalization only happens at the very end of the class, after the actual final grades. I just add up all of your exam scores. And then I add up all of the other people's exam scores. And then I start to normalize at that point. So that's where that stands. I tried to upload your clicker questions over the weekend. For some reason, it didn't want to go from my computer at home. So I'll try and do that as soon as I get back to my office after class. Yeah, Ian.
Are you proud of us?
Am I proud of you? Some of you I'm very proud of. Some of the rest of you I wonder what my teaching skills are. So, yeah, how does one balance that? I don't know is the answer. Okay, so that being said, once I actually figure things out where to normalize from, that's where we'll do the normalization. It will not be 46, I promise. So probably not a 45 either. Okay, so let's move on. I know everyone would much prefer to talk about the exam rather than the subject matter, but a couple of things that I wanted to point out here. We'll sort of finish up the stuff in Chapter 6 and then move on to Chapter 7 today. And someone asked me on, I think it was email, when we're going to talk about CRISPRs. Probably the third to last lecture. So when we start to talk about techniques, we'll probably talk about CRISPRs. I still need to figure out exactly what we're going to do as far as the rest of the curriculum is concerned. But just a couple of things sort of following along with translation. Translation is absolutely critical to cellular life as we know it. And so it's not terribly surprising that most antibiotics that work for actually both eukarya, bacteria, and probably for archaea, although there are no archaeal pathogens, so people haven't looked into them too much, are acting on the ribosome. And so just all of these different circles here. No, I don't expect you to remember all the names here, tetracycline, streptomycin. There's so much resistance anyway to these that they're not used terribly often. But a lot of modifications on these. And again, the basic story is really about the ribosome, and it's taking advantage of the differences between the bacterial ribosome and translation machinery and the eukaryotic ribosome and translation machinery. If you think about taking an antibiotic to treat a bacterial infection, if you can get something which is specific to bacteria, that's going to be a really useful tool. And so that's what a lot of these guys are here. They're specific ribosomal, and then, interestingly enough, this one RNA polymerase here, again, not surprisingly, completely conserved in all these organisms, but different enough between eukaryotes and bacteria that you can treat with a particular drug that will just kill off or at least slow down the growth of bacteria and not do anything to the human cells. Of course, now we're realizing that with all of our microbiome that a lot of those are good bugs too. So if we're killing off the good microbes as well as the bad microbes at the same time, maybe it's not such a good thing. So once we finish with translation, now all we have is a string of amino acids. That string of amino acids doesn't have any structure, so no structure, no. Is that, you're more awake than I am this morning. So one of the really fascinating things that was found, I don't know, probably about 30 years ago, is that you can take just a stretch of amino acids here, it's come off the ribosome, but also you can take a protein that's already been formed normally, has a nice structure and a nice function, and completely pull it apart. It will go back together again and get the right function back, which is really pretty amazing that you can, pull this whole thing apart and then it will come back together and give you that same structure and function again. It's not absolutely the case. You can't do this with all proteins, but for the most part, you actually can. And the way that this works seems to be mostly you make some secondary structure, and then the secondary structures come together to make your final tertiary structure. And then if you've got other protein subunits, you'll make a quaternary structure as well. But one of the things that people have found in studying this, what people call the protein folding problem, is that very often you have an intermediate in your protein folding called the molten globule. Now, what the heck is a molten globule? Basically, a molten globule is a form of your protein that has most of the secondary structure, but very little of your tertiary structure. And remember, tertiary structure has to do with all the side chains interacting with each other, whereas the secondary structure is all about the backbone amino acids, the atoms that you have in every single amino acid interacting with each other. So alpha helices, beta sheets, etc. They're all about those side chain amino acids. So it seems that these main chain interactions happen really quickly, and so you end up with secondary structure, but getting to your final tertiary structure takes a while. And so this is just an example of a so-called molten globule for one protein here. This is its final form. They probably look really, really similar to each other, particularly if you squint. But there are a couple of differences. One is this alpha helix, which was not complete here, and that everything is packed together a little bit more tightly. This is now very stable. This is much, much less stable, but it happens really quickly. Now, if you think about the number of different amino acids, 20, and the number of possible interactions they could all have with each other, you end up with an absolutely ridiculously astronomically large number of possibilities on how all of these things could interact with each other. So it seems that secondary structure is easy, tertiary structure is hard. Not just hard for us to predict, but it's also really hard for the cell to do. In some cases, this tertiary structure happens without any trouble at all. And so the co-translational folding, and this is actually a lot like what we saw in that animation with translation, where the little red peptide was coming out of the ribosome and sort of folded into alpha helices and structures and then popped off. This is actually the minority of proteins that do this. But you have your polypeptide chain makes all of its secondary structures, but also tertiary structures. And then when it's released from the ribosome, because it's got to all of its release factors, et cetera, this protein has its full form and function. So this is perfectly fine. But again, that's usually not the case. Usually you end up with one of these partially formed proteins, so it's got some secondary structure, but doesn't have its final tertiary structure. So then what do you need to do? And I actually love this terminology for chaperones that are helping proteins to fold. Well, actually, I don't see them so much as helping the proteins to fold, but as the father of two daughters, I think about they're preventing the wrong kind of interactions, which is exactly what chaperones are supposed to do, is preventing these wrong kinds of interactions. So that seems to be exactly what these chaperones are doing. They're making sure that the right interactions happen, but that's mostly in terms of stopping the wrong kinds of interactions that happen. And so this is sort of the cartoon message, how this works. Really quickly, you get these molten globule formations, and then with the chaperones, it sort of says, now, maybe I don't like that particular interaction that's going on here. We'll try it again. Oh, eventually, this one could be an OK in-law. We'll deal with that. On the other hand, if these are not properly functioning, then they get put into the garbage disposal, and we'll talk about that in a little bit more detail. So one of the questions in terms of how all of these chaperones were discovered, of course, in the study of virology. You know, obviously, that's where you learn all of these things. But it turns out that this particular, and particularly the HSP60 family, were found because they were mutants that didn't allow the formation of certain viruses. And so that's where this GROW comes from, GRO, for the growth of a particular bacteriophage. And so if in this case, E. coli had a mutant in that particular gene, the virus couldn't grow, and we found out that's because the virus couldn't get itself put together, so it couldn't assemble properly. So that's where GROE and that family of the HSP60 comes from. These are also known as heat shock proteins. Why heat shock proteins? Because if you heat up any cell, it turns out, and even our favorite organisms that grow at 80 degrees Celsius, if you heat them up 5 or 6 degrees, they also will have these heat shock proteins that get expressed. So it's the whole range of organisms. Even organisms that grow at 100 degrees Celsius, you can heat them up another 5 degrees, they'll turn on a heat shock response, which is really pretty amazing. Now, what is a heat shock response? The heat shock response, just a sec, is certain proteins that are made much more than they otherwise would be when there's a change in temperature that happens. Yeah?
Is that like an early combative method for like thermal vent life, like to deal with extreme temperatures?
So the question has to do with whether this kind of, call it heat shock response, is important for some of these hydrothermal vent microorganisms. The answer to that is not entirely clear because there haven't been very many mutants which have been made, unlike the case for E. coli, where you can look very carefully and see if you knock out some of these genes, the cells then die if you go to those higher temperatures. That hasn't been done for a lot of these hyperthermophiles, unfortunately. Love to. Great experiment. Do it. And the thought is that these heat shock proteins then will help again, just like we talked about before, stop the bad interactions from happening. And the bad interactions mostly seem to be hydrophobic parts of your protein that are interacting with hydrophobic parts of other proteins. And what happens then is these proteins then precipitate. And those precipitation is really bad. We know about it very well in terms of a lot of the neurodegenerative diseases. A lot of these are precipitation of proteins. And the same thing is true in E. coli. Yes, they don't get neurodegenerative diseases, but protein precipitation in E. coli can be a real problem as well. So what do these chaperones look like? There are two sort of main classes of chaperones, and they're, again, it's a total oversimplification here, but the GROE set of chaperones, also known as HSP60. HSP just comes from heat shock protein. 60 is the relative molecular mass of that protein. So it turns out that about 60,000 Daltons, which is approximately the molecular mass of a hydrogen atom. We can talk a lot more about that later. But about 60,000 Daltons. And then there's a whole other family, which are HSP70s. And I give you one guess, their size. 70 kilodaltons, exactly. And those were found, again, because people were just looking at proteins that were being overexpressed in E. coli when they went to this higher temperature, and found in pretty much all organisms. Bacteria, archaea, eukarya, you name it, have these HSP60 and HSP70-like proteins. So HSP60, this is one of the very first ones to be discovered. Again, it's GROE, and it turns out GROE-L and GROE-S. There are two different proteins that are involved there. One is 60 kilodaltons, and the other one is considerably smaller, i.e. the S that goes with it. What seems to happen with these HSP60s is you have a not completely folded protein that comes into this cage of infinite dilution, which is what some people, again, this is the other terminology that people have used. But basically, I like to think of it as you put it in your shaker, you put the top on, you shake it around a little bit, and hope that the right interactions form. And that shaking process also has ATP hydrolysis. So what's happening is it's binding to the outside of this cavity on the inside, going back and forth until you finally have a properly formed protein. The top comes off, and you have your margarita, daiquiri, whatever you want to have at that point afterwards. So this is the process, again, that's happening with HSP60s. We know a lot about the structures of these. We actually have high-resolution structures for HSP60s from lots of different groups of organisms. Yeah?
The ATP is to cap it or to shake it?
The ATP seems to mostly be for shaking and then taking the cap off at the end. So that's where the energy is. On the other hand, there's also this whole other set of proteins, the HSP70s, which seem to act much earlier on in the whole folding process. So that's what's down here at the bottom. HSP70s, these little blobs are way too small. I'm not quite sure why they made them so small in this image from our, our textbook. But here, HSP70s associate with these peptides right as they're coming off of the ribosome, and basically, again, seem to serve this classic kind of chaperone machinery, keeping away the bad stuff and allowing the good stuff to happen. And here, ATP hydrolysis mostly seems to be involved in getting the HSP70 off of this finally properly formed protein. So there are lots of other chaperones, larger molecular weight, smaller molecular weight, you name it. But these are the two main ones that do seem to be very important for protein folding. So we've got those that fold just as they're being made from the ribosome. These are the ones right here, molecular chaperones. And then we have a few that just don't work properly. This might be because, for instance, we had a broken messenger RNA and some of the degradation mechanisms that we talked about before the midterm, which we've all forgotten because that was the last midterm. And these are things that then are destined for the garbage disposal. So what is the garbage disposal? The proteasome. So the proteasome is one of the most common proteins that you find inside pretty much any cell. And it's this, actually looks a lot like my garbage disposal at home. This cylindrical structure which has lots of proteases on the inside of it. And they're pretty non-specific proteases. They'll chew up pretty much anything that gets into it. Now, because they can chew up pretty much anything that gets into it, and there's a lot of it around, you need to be careful about what gets into the proteasome in the first place. And so all the proteasomes also have these regulatory proteins that are attached at either end, and these are basically the gatekeepers. They say, okay, this protein needs to be degraded and chewed up. This other protein does not need to be degraded and chewed up. And for the most part, at least in eukaryotic systems, we've got polyubiquitination, which is the signal to say, hey, this protein needs to be chewed up. And we talked about ubiquitin ligation. We'll go back and cover it again really briefly here. But if you've got polyubiquitination, which has happened on a particular protein, that's going to bring the protein to the gatekeeper right here. Then, once it's bound, the ubiquitin is removed, and the protein itself is basically unwound and poked into the garbage disposal where it gets broken down. So there are actually two proteases here, two kinds of proteases. One which cuts off the ubiquitin. So this is a very specific protease, and it happens up here in this regulatory subunit. And then down here are all of the non-specific proteases. They're going to chew that amino acids polypeptide chain down into short, either couple of amino acids or single amino acids, which can then get recycled by your amino acyl tRNA synthetases, et cetera, throughout the whole process. Turns out that proteasomes and this whole degradation process are really important for proper cell growth. And so there are a lot of now drugs, particularly chemotherapy drugs, which are trying to address the proteasome. So if you get too many of these unfolded and bad proteins, it can be a problem, and then the cell will end up going through apoptosis. So this is actually a really interesting drug target as well in terms of thinking about messing with how cellular function is happening. Just wanted to remind you a little bit that ubiquitin is not just for degradation. And we talked about this last time. In fact, it's exactly the same slide that we showed you before. This polyubiquitination that happens here, this leads now to the proteasome, but you can have all kinds of other ubiquitination that happens which seems to serve as signaling molecules. How do you put ubiquitin on? Again, we've talked about this before, so I'm going to go over it pretty quickly. You have a ubiquitin activating enzyme, E1, adds ubiquitin to itself. That gets transferred to E2. And then it's E3, which is determining the specificity for your final protein, the protein that's going to get this polyubiquitin that's been added to it. It can be something which is important for signaling, but also could be all of these polyubiquitination that happens saying, hey, this is a protein that needs to be degraded, improperly folded, etc. Yeah?
It was called ubiquitin ligase. You described it more as a scaffold protein than as a true enzyme, right?
So, some people remember too much. So I actually went and asked Dr. Singer about that. So the question here is, is the ubiquitin ligase really a ligase? And I asked him about this because he works on E3 proteins in particular, and he says yes and no. So some actually do seem to have that enzymatic activity. Most of them don't, and most of them just seem to be serving as adapters. Yeah?
So the ubiquitin ligase is E2 and E3?
Yeah, so usually when people think about this, they think about the 2,3 as being the complex, which is the ligase. But sometimes people talk about the E3 specifically as ubiquitin ligases, and I think that was getting back to Ian's question, is that E3, in some cases, the E3 actually does have some enzymatic activity. Usually it's the E2 which is doing that. So that's probably a really good way of thinking about it. E2 plus E3 is definitely ubiquitin ligase. Thank you. Okay, so how do you get these degradation signals? Obviously, if you've got this extra hydrophobic piece, again, your molten globule that hasn't had the right kind of interactions, hasn't had the help through the chaperones, it hasn't folded up as it's coming off of the ribosome, then you're going to get polyubiquitination and degradation. But as some of you may have heard about in cell biology, if not, you will hear about in cell biology later, there are lots of proteins in the cell, particularly those that are involved in the cell cycle, which are really unstable. And we talked about those when we talked about eukaryotic replication origins. Again, way back, like 6 weeks ago or something. So these are things which get ubiquitinated and get degraded. And for that reason, they're actually being regulated in terms of the amount of protein which is around. And we'll talk more about that when we talk about regulation a little bit later on here. So you've got lots of different ways that you can have this happen. Again, we've talked about it before. It's all about change of structure. So you can have a change of structure due to kinase activity, dissociation of different proteins, or some cases actually even cleavage that takes place of your particular protein. So this is all that we have talked about basically so far. We had replication back up here, and then the last set of lectures looking at transcriptional regulation, where you have a promoter. The RNA polymerase makes a copy of the RNA, which has got introns and exons in it. Capping and splicing happens. While this is going on, you have polyadenylation, and polyadenylation, at least in eukaryotic cells, is very closely connected to transcriptional termination. We have export, messenger RNA degradation, translation, and as we talked about today, protein folding, and then in some cases, protein degradation. So all of these steps, the transcription, the capping, elongation, and splicing, termination, export, degradation, translation, folding, and degradation are all regulated. And so that's what we're going to spend most of the rest of the class talking about, is all of those regulatory steps. But before then, what do? Clicker questions. Thank you. So here, which of the following is most likely to have a direct protein, I should say protein-protein interaction or protein-RNA interaction with the ribosome? HSP60, HSP70, the proteasome, E2, or E3? And we're going to get 100% sometime. I'm going to try. 10, 5,
Unless somebody just doesn't care, they just click something. 25% or 20% chance.
Okay, let's look at our answers. So, not 100%, but most of you seem to be on the right track here. Why not HSP60, E2, E3, or the proteasome? Because these are things that are happening after you've had production of that molten globule. So particularly the proteasome is degrading unfolded proteins, and the protein hasn't had a chance to fold yet. That would be a really bad idea. If you're ubiquitinating something right as it's being made, also probably a bad idea. HSP60, to get into that bottle of infinite dilution, you actually have to have been released from the ribosome, because your ribosome is not going to be poking it in during that process. So we basically only have HSP60, HSP70 as the option. And of course, it's also, as these things are being made, we have that interaction. You had a question in the back.
Does HSP70 always interact with proteins coming off the ribosome, or is it selective?
So the question here is, again, just repeating it so it's on tape, is, does HSP70 always associate with the ribosome, or is it sometimes not associated with the ribosome? We haven't talked about it, but it's sometimes not associated with the ribosome as well. And so you can have HSP70s floating around freely in your cytoplasm as well as being directly associated with the ribosome. So you can have both. Okay, so now switch gears, talk about regulation, although it's a really nice transition to go straight from what we just talked about, transcription, all the way through protein stability. Regulation is almost all about the DNA, and particularly the transcriptional process. And when I say almost all about the DNA, this is something that I added a couple of years ago, because RNA is clearly also very, very important for this process, and a lot of small RNAs as well. We'll talk a little bit about differential regulation. This really has to do with the fact that not all genes, or particularly gene products, you don't want to have all of those all the time. And how you can do that, since practically all the cells in our body have exactly the same genetic content in terms of the DNA. Very different, however, in terms of the proteins, and even the activity of the individual proteins in the different cells. So we'll talk a lot about that. And then more specifically, why I brought my model today, we'll be talking about DNA binding and the various different ways that you can have proteins interacting with DNA. And kind of make a long story short, it's all about the major groove. Surprise, surprise. And then a lot of these DNA binding proteins are dimers, i.e. they've got two separate polypeptides that come together. Turns out to be extremely useful for specificity, but also in terms of how you regulate individual genes, how you can turn something on and off and have much more of a switch rather than a long partially on, partially off kinds of process. In these DNA binding proteins, we've got lots of motifs. We barely touched motifs before. These are separate parts of your protein. They're not domains. They're just a little piece of your protein, and they don't have a structure in and of themselves, but they can give you some hint in terms of the function. So we'll look at that. Cooperativity is another reason you want to have dimers. Again, this is on-off switch rather than a sort of a linear kind of progression between on and off. Makes things a lot easier in terms of regulation. And then if we get there, we'll talk about nucleosomes, but at this rate, we're probably not going to. So this is just, again, repetition of what I mentioned before. Sort of the almost extreme examples of different cellular morphologies, but identical DNAs are between liver cells and nerve cells. So this guy's got the same DNA as this guy, or this whole guy here, but clearly very different in terms of their structure, very different in terms of their function. So how does that happen? It's not because they've got a different DNA content. It really has to do with what proteins are made and how they're responding to various different proteins. And this is very much an overview for the next 4 or 5 lectures now. So we'll talk a lot more about it. Why is DNA sufficient? Well, we've all heard about Dolly, hopefully, the cloned sheep. Well, Dolly came on the shoulders of lots of other organisms, particularly frogs. Never would have thought of sheep standing on frogs' backs. But these were the very first experiments which were done to show that you can take a nucleus from one organism and transfer it into a cell that's been enucleated or destroyed in some cases, and then still get development, but you get development based on the nucleus and the DNA, not based on the rest of the cell that it comes with. Yeah?
This sounds a lot like the 3-parent project that's going on. Is that true?
So, sounds a lot like the tri-parental project, which is happening right now. So, it's getting a little bit of an aside, but I wasn't going to mention it, but we're happy to now. So there's a group at OHSU at the Primate Center. Metalipov, I think, is the PI who's working there. He's very interested in these kinds of processes, and it turns out that there are a number of mitochondrial diseases. And mitochondria, these are the power-making parts of your cells, which also have their own DNA in them. And sometimes there are mutations in those DNA. That actually comes from this cell down here, the mitochondria. And so what they're looking at now is this nucleus, of course, is a diploid nucleus. And so you can actually make a diploid nucleus from 2 parents and then have a donor parent which has the mitochondria here. And so you'll get that nucleus from the usually in vitro fertilization and then put it into the other cell. And that's how you do your triparental offspring. And it does seem to be working. They've done it, again, at the Primate Center with lots of non-human primates, and they're just starting to do it in the United Kingdom. I'm actually using this for people because these are really debilitating diseases, all these mitochondrial diseases. Basically, the child would never make it. Okay, so first done with tadpoles. Turns out you can do the same thing with plants, and everyone's favorite, of course, is Dolly and Polly, who we've gotten rid of over here. No, should be anonymous. That's probably why. So the idea here, again, is taking a nucleus from one cell, a donor here, and then putting it into an egg, which actually ends up with multiple nuclei here, and there are ways to get rid of it, and then these embryos which are formed. So why should anybody care? Yes, you want to have your cloned cat or your dog. But anyone know why they tried to do this in the first place? Other than it was really cool science. So had to do with actually producing pharmaceuticals in milk. And so a lot of pharmaceutical proteins are really hard and really expensive to make. Milk is filled with proteins. And so if you can actually make a transgenic, particularly sheep in this case, which is now expressing some of these proteins in its milk, then it's really easy to purify those proteins, get a lot of them. And so this whole cloning process was actually about trying to make pharmaceuticals in a cheaper way. And once you've spent all this time and effort making a particular organism that can produce a bunch of this milk, well, they die after a certain period of time. So having a clone which has that same DNA is the reason that they did this in the very first place. So should we be doing this with humans? There was actually a really interesting discussion that I think came out about a week ago. I'll try and post it on D2L. The National Academies of Science and Engineering and Medicine got together and put out a report and said basically, under most cases, no. But getting back to your question about the whole triparental thing, if there are some kinds of really clear genetic diseases which are there, then we might think about doing some of these things and doing some gene editing. Turns out that now we're doing a different process for that rather than doing this with humans here. But it has been shown it does work with human cells. You can do this. The main problem is that in the case of Dolly, I think it took about 400 of these different transformation processes to actually get it to work. So it's pretty low yield. It's really hard to do these kinds of things. And then there are real issues about trying to do this with human eggs as well. So yeah, it's just the DNA, but what happens to that particular DNA? Well, again, what we talked about last time, last set of lectures. A lot of it's about transcription. And the transcription can be either extremely similar in lots of different cells or very different, as we'll look at on the next slide. But first, I wanted to talk a little bit about how people look at amounts of transcription. This is something also that has changed in the about 15 years that I've been teaching this class. This was basically impossible to do this experiment when I first started talking about this stuff. And so, excuse me, what we're looking at here is RNAs, and this is actually really small RNAs. They're maybe about 100 nucleotides or so. But looking at all of the RNAs that are being made by any particular cell at any given time. So you just take all the RNAs out of a cell, and what you do is you sequence all of them. And then you just look at where these sequences match relative to the DNA that you have back up here at the top. And so one of the things that was seen pretty quickly, just pulling the RNA out, not surprisingly, most of those RNAs are in the exons. You've got a little bit of RNA left from the introns, but it's mostly in exons here. And this is literally millions of sequences. And it's all done in parallel, and we'll talk about how that happens again right towards the end of the class, second to last lecture probably. But literally millions of RNA sequences that you get, and in the computer line them up with the genome, because we now have the human genome sequence. And so any place where you've got up here certain number of sequences, down here a lot smaller number of sequences. A couple of things to see here, though. Even though you're pulling out all the RNA, which introns should have been degraded, there's still quite a lot of that intron RNA which is around. But the main thing to look at here is that all of these different cells, embryonic stem cells, liver cells, muscle cells, etc., are all expressing this particular RNA and presumably this protein as well. But what about down here? Dr. Stedman, that's not an intron. Why are you getting all this RNA? Well, that's about regulation, and we'll talk about that a little bit later on. So the flip side of this is now taking a gene which is expressed only in very few cell types, and basically do exactly the same thing. Take all the RNA out of that cell, sequence the ... out of it, and then look where all those sequences are. And if you just look at the region of the genome, which is this tyrosine aminotransferase gene, you only see exons from this gene in liver cells. You don't see them in stem cells, you don't see them in muscle cells, you don't see them in any of the other ones. Now, there's a little bit of background, a few sequences that you see here, but basically that seems to be either background transcription, or it might also be a small RNA which is being made in some of those cells that actually has nothing to do with this gene up here at the top. So RNA transcription is highly variable between various different cell types. Some cell types make some genes all the time. Actin is really important for cellular structure, whereas tyrosine aminotransferase turns out to be really important for metabolism, which is going on inside the liver. But RNA is all well and good, but is that really what's happening? Okay, in some cases we've got active RNAs, but it's really the proteins which are doing the job. Now the problem is proteins are a lot harder to identify. Sequence, we got the human genome sequence. We can look at RNA. We can match it exactly. That's really easy to do. Looking at all the proteins is a lot harder, and we can't do millions of sequences and compare them all to each other. So that brings us to proteins or proteomics. This actually is only a very small part of the various different proteins which are being made. But again, in a excuse me, identical cell, you take all the proteins out, you look at all those proteins, and then you compare all the proteins which are being made there to proteins that are being made in a different kind of cell. And so here we're just looking at brain and liver. All the red dots are the same protein. All the different dots are, sorry, all of the proteins which are different are now in blue. Now, how do you separate all these proteins relative to each other? This is called gel electrophoresis, and we'll talk a lot more about it later. But what we're doing is we're separating based on size, but also based on the overall charge of the protein. And you have to do what's called these multi-dimensional kinds of separations. Otherwise, you would never be able to separate all of these proteins relative to each other. And one of the things you can look at over here, there's a protein that's got exactly the same size, but lots of different charges on it. And one of the things that that could be is phosphorylation, different numbers of phosphates. That's going to change the charge that your protein happens to have. So one dot on here doesn't necessarily represent one protein. It's one potentially modified protein. So you've got literally thousands of spots here, and then you can sort of try and compare them with each other. And even then, each of those spots, is this one active with this number of phosphates on it, this one active with this number of phosphates? We still really don't know. And so it's getting to be, very challenging. We know the genome sequence. It's great. We've known the genome sequence actually for over 10 years now. Do we know what the proteins are doing for 90+% of them? We really don't have a very good handle on it. So it's that end which is difficult to look at. So again, this is just basically exactly the same slide that I had right after the translation discussion, just turned 90 degrees here. So different kinds of regulation, different places that you can have control that happens in going from the DNA, which again is all you need, just so you're cloning, you get the nucleus, you put it in, that's what you get, over to actual phenotype, what's happening, that's all the way over here with your final active protein. So first kind of control, and we're going to spend most of our time talking about this because this is the first step. What gene gets turned on? What gene gets turned off at any particular time? Why is this so useful? Well, because you don't have to do all these other things. If you haven't turned the gene on, you're here. You haven't spent all of this energy. However, once you've turned on that gene, then you can have different kinds of processing. And we talked about alternative splicing before. That can give you a slightly different protein. Turns out to be really important for different cell types, and we'll talk about some of the modifications that happen there. RNA transport and localization. Turns out this is really critical for development. Is anybody taking developmental biology? No? No? Yes? You're supposed to take this course first. But developmental biology, a lot of where RNAs are in an embryo turns out to be extremely important for how development takes place. mRNA degradation. We talked about this for malformed messenger RNAs, but it turns out that this is also heavily regulated. So if you have a particular RNA in one cell, you want to make a whole bunch of protein from it, you want that to be very stable. If, however, you have an RNA that you don't want to make a lot of protein from, you want to have it being very unstable. So we'll look at that. We talked a little bit about translational control already. A lot of that's going to be the extra piece that I talked about, the 5' untranslated region, when we looked at all those RNA sequences. And then our activity control. This can be in terms of degradation that we talked about, or particularly modification. So it can be GTP hydrolysis by your G protein, changing the structure. It could be your kinase, phosphatase, changing the structure by adding on this particular modification that goes with it. So this is going to be your fastest way of changing activity, changing your gene expression. This over here is going to be your most efficient way of doing that because you'd have to do all of the other things. But it takes a lot longer. If you're making your RNA, got to process it, got to export it, got to translate it, etc. So any kind of change that happens at a transcriptional level is going to be a slow change, but means it's going to be relatively energy efficient. On the other hand, this change here will be fast, but you've had to have made everything beforehand in order to be able to have this kind of regulation. Questions, comments, worries? Everyone grab their clickers. Yes, good. So which of the following kinds of gene regulation is most likely to be the most energy efficient in terms of NTP hydrolysis, NTP, GTP, ATP, etc. Transcription control, mRNA processing control, RNA transportation control, translation control, protein activity control. And I didn't turn it on until a while, so make sure you vote early, vote often. Five. So close. Everyone's still trying to mess with me here as far as this is concerned. So in terms of your NTP hydrolysis, every nucleotide that you make is going to have a hydrolysis of NTP. So anytime you're extending something, it's going to be using your NTP hydrolysis. All the rest of the stuff is happening downstream. In terms of your protein activity control, that is true that that individual change is probably not going to use a huge amount of NTP hydrolysis, but in some cases it's actually none whatsoever. But you have to have made the protein in the first place. And making the protein in the first place, you've got to transcribe the gene, you've got to do all the processing, you've got to do the translation, and translation is really where you have a huge amount of NTP hydrolysis, your GTP hydrolysis for each of the translocations that takes place, the checking to make sure that you have all of the appropriate interactions, and charging your amino acids in the first place. So yes, it is A here. Okay, so now finally get to my model. Transcription factors. Somebody asked me a question about sort of general transcription factors versus your specific transcription factors. Unfortunately, when most people talk about transcription factors, they usually mean specific transcription factors here. These are also known as your gene regulatory proteins. About 10% of the genes in our genome seem to encode these gene regulatory proteins. Again, so not terribly surprising that that's where a lot of regulation is going on. It's going to be proteins interacting with your DNA. 90+% of these interact in the major groove of DNA. It's an intact double-stranded DNA, and that's because that's where the information is. If you're going to bind to a particular and now specific DNA sequence, it's going to have to be interacting in some part which is clearly specific. These are also known as cis regulatory sequences, cis versus trans. Cis right next to, trans further away. Everyone remember that from Gen Chem? Yes? No? So we'll keep using the same kind of terminology. So cis-acting sequences, nearby. And nearby is a relative term as far as molecular biology is concerned. Turns out that a binding protein nearby to something that's regulated can actually be tens of thousands of nucleotides away. So, but all still relatively nearby. Now, why are we binding in the major groove? It's because, as we looked at before, that's where all the information is. A protein can read what that base pair is just by the individual atoms which are present in the major groove. And again, I'm not expecting you to remember all of the different donors, acceptors, etc. But again, it's all about the major groove. There are some things in the minor groove, but this is going to be much less specific in terms of the interactions. How do you interact with these things? Lots of different possible interactions, but the main ones have to do with hydrogen bond interactions. So these are just two of the amino acids that can make hydrogen bond interactions, asparagine and arginine. Just looking at here, regular base pair, so this is our AT base pair, has its hydrogen bonds. So here's our major groove of DNA. And now we have an asparagine, which can also make hydrogen bonds here in the major groove. Arginine can make hydrogen bonds to GC base pairs. These are just like we talked about way back when, lecture 2, I think. It's all about these weak interactions and many, many, many weak interactions. The problem, unfortunately, with these kinds of interactions, like, oh, cool, look, an asparagine, it binds to an AT base pair. Look, an arginine is going to bind to a GC base pair. Well, unfortunately, these things can bind all over the place. And so it's not always going to be a nice sort of, asparagine, AT, arginine, GC. It's really all over the place. And so one example of that is just looking here, where we have lots of different amino acids interacting with a relatively short piece of double-stranded DNA sequence. All of these different amino acids are interacting with all of these different base pairs, and some even in the minor groove as well as in the major groove. Some interact with the sugars on your backbone. Of course, this is not going to be a specific interaction. The specific interactions are all going to be with the base pairs. But because of this, it's practically impossible to predict, just given your nucleotide sequence and your protein sequence, how they're going to interact with each other, or even if they are going to end up interacting with each other. So this is one of the big puzzles of molecular biology, is to try and figure out what structure in protein is going to interact with what sequence in your DNA. Yeah? What are all the different numbers here? Like asin 51, for instance, here. So this is the 51st amino acid in this particular protein, which is an asparagine. Here's arginine at position 53, tyrosine at 25, lysine at 57. And what this is telling you is that that protein has a three-dimensional structure. So all of these side chains coming from various different parts of the protein are all now interacting with the DNA. But it's really hard, again, to predict what those are going to be. And that's basically all that this is about. Now, you can make some predictions. And these predictions are what we're going to be looking at on the next couple of slides. These are motifs. So these are parts of your protein that are going to have a function, but not by themselves, only in the context of a complete protein. And one of the best known of those is what's called the helix-turn-helix motif. And what is it? It's a helix, alpha helix, with a short stretch of amino acids, and then another helix. But what's really important about these helix-turn-helix structures is you can guess, based on your amino acid sequence, that it's going to have a particular kind of secondary structure. And actually, secondary structures can be predicted reasonably well. Tertiary structures are really hard. But secondary structures you can predict pretty well. So if you see a predicted helix with a little short stretch of amino acids between them and another helix, that could be a helix-turn-helix. And these helix-turn-helixes are great in terms of DNA binding because they've got one helix that sits down in the major groove of the DNA, and the other helix that sort of sits across it and holds it in there. And so here, and this is known from a lot of different DNA binding proteins, the so-called recognition helix, this is the one that's got amino acid side chains, again, sticking out, because that's what alpha helices do, and interacting with the DNA and being held in there by this other helix of the helix-turn-helix. And that usually has non-specific interactions with the backbone. So it's really literally holding that helix into the major groove of DNA. Now, why do we know about this? Because there are lots and lots of helix-turn-helix containing proteins that we know are DNA binding proteins, and we actually have high-resolution structures for a lot of those. Yeah, you had a question?
Yeah, about the recognition helix, it makes a specific binding to it, and then it makes a non-specific binding to the backbone?
So the recognition helix makes specific. It's the other helix which has the non-specific interactions. And here, conveniently, red is the specific one, blue is the non-specific one. Yeah?
And that can be changed by processing mRNA differently, like you can combine a recognition helix to a very specific portion of the DNA, and then if you have like a repeated sequence of mRNA that just holds it, like it anchors
So the question here basically is, getting a little ahead of ourselves here, but it's a great question, is, can some of this be potentially why you have alternative splicing? Now, usually the alternative splicing has to do with different domains, not so much different motifs. But definitely what you can do, and there's nice examples of this, is bring together two different, usually, DNA binding domains with each other, and then get a different specificity. And we'll talk a little bit more about that later. Turns out that the helix-turn helix, one of the things that you may notice down here is that lambda is one of them. All of these proteins are bacterial proteins or bacterial viral proteins, which usually don't have very much splicing involved in them. And these are, again, of these structures which we know about. We're going to talk a lot more about the tryptophan repressor and the lambda repressor as we move along here. These are classic regulatory proteins, bacterial regulatory proteins. But one of the things which is really obvious here from all three of these different structures is that they have these recognition helices that are almost exactly 3.4 nanometers apart. Why? 3.4 nanometers happens to be almost exactly the distance between adjacent major grooves on your DNA. So if you've got one recognition helix that's sitting here, the next recognition helix can sit right here. So all of your protein is going to be sitting on one side of your DNA. And this is extremely common in bacterial regulators, also quite common in the eukaryotic regulators. We have these proteins, and unfortunately, these are actually pretty much the scale over here. The proteins relative to the DNA, where we've got these alpha helices sitting down in adjacent major grooves, but you'll notice that the rest of the protein is really quite different. These alpha helices, again, are all about the same distance apart, but the rest of the protein is really quite different. And that's, again, classic kind of thing about protein structure. Most of your protein is just involved in getting a very small part of it to have the right structure and eventually have that right kind of function. So why have these two alpha helices in adjacent major grooves relative to each other? One reason is if you just look at the DNA here, you'll notice there are only about 5 or 6 base pairs that this alpha helix can interact with. 5 or 6 base pairs in even a genome which is as small as E. coli is not going to be completely specific. At random, you're going to find that particular stretch of 5 or 6 nucleotides quite frequently in the rest of the genome. However, it's a lot less likely that you're going to find that stretch of nucleotides right next to another stretch of nucleotides. And so that is probably why we've got a lot of these dimers, which are binding to specific DNA sequences, and particularly dimers that bind in a either head-to-head or tail-to-tail interaction. What do I mean by that? Well, what that means is these are binding to inverted repeat sequences. Everyone remembers our inverted repeats and direct repeats from something that happened on Friday. So on one strand, your inverted repeat, let's get my pointer to work here, is going to be 5' AACAC, and then on the opposite strand, 5' AACAC. So the protein here, your recognition helix, binds to this particular sequence, and this one binds to this particular sequence, but in opposite orientations. They're inverted repeats relative to each other. And now we've got our DNA binding motif, and now these guys are interacting with each other through a dimerization motif. And we'll look at dimerization motifs here a lot more in just a second. So here we've got one protein, but as long as you've got 2 of them that are interacting with each other, that one protein just by itself would only bind to 5 nucleotides. That's not specific binding. But as a dimer, it can now bind to 10 nucleotides, and that's going to be extremely specific binding. Yeah?
So you talk about the genome and how much of the DNA is regulatory. What you're talking about is the interaction between the proteins that will either signal or prevent or enhance transcription of that DNA. Is this what regulatory DNA means? doesn't actually code for anything, it's just a binding site?
Okay, so thanks for finishing up the question there. So basically the question is, what about regulatory DNA? Or maybe, to paraphrase the question, what about functional DNA potentially? So there are two different things. So when I talked about the 10% of our genome, which are these regulatory proteins, those are right at the beginning we talked about regulation. That 10% are the 10% of the protein coding genes which do this kind of thing, like the green thing here. Then there's also a lot of DNA sequence which interacts with these particular DNA binding proteins. And that's a separate set of DNAs. When we talked about the human genome and all the functional sequences which are in the human genome, a lot of those functional sequences are going to be these kinds of things that you have proteins that are interacting with. Yeah?
Yeah, I have a question about when it binds to the dimers. How does it become more specific when it can bind to more?
So if you just had a monomer, it would just bind to, okay, we're going down here, 5' AACAC. And that at random, just if you think about 4 nucleotides, five actually in this case, 5 to the n is that's 1 every 512 nucleotides. Just at random, you'll have that sequence. And so that could bind potentially randomly in a bunch of different places. However, if you've got 2 sets of 5 nucleotides, which are going to have that specific sequence, that's going to be 1 in actually over 1 million different places that can now bind to. And so it's a specificity because you've got these 2 things that have to be right next to each other. And so that's the key of having the dimers. They have to be right next to each other. If it's just a monomer, they could be all over the place. Okay, so those are the classic DNA binding motif in bacteria is the helix turn helix. Eukaryotes have a few more, and I actually like to think of this one as sort of the eukaryotic helix turn helix motif, also known as a homeodomain, and you can tell immediately by the name it's a domain, so this actually does have its own structure and can just be this particular protein. We looked at one of these a little while back. So this actually does have a particular structure instead of just having a recognition helix here in red and a second helix which goes on top of this. This one actually has a 3rd helix, which is part of it, and part of this helix reaches over and interacts with the minor groove of your DNA. So here, recognition helix, very similar to what you see in bacterial systems, but this extra helix, it also has some interactions in the minor groove. Yes?
So for eukaryotic versus bacterial, since it's kind of hard to keep track of all the study, it's always going to have an E in front of whatever it is when it's eukaryotic versus bacterial.
Okay, so the question here, sorry to interrupt you. Is that done? Yeah. So the question here is, what about my terminology here? I think I'm one of the very few people to actually use this terminology for homeodomains. I find it easier to think about the structure if you think about it as being actually really similar to helix turn helix, which is what you find in bacteria. But homeodomain is what you find in eukaryotes. 99.99% of the population will just call it a homeodomain. They won't give you that nice little extra E. I try and give it to you to say, this is a similar structure, but it's in eukaryotes. But this is my sort of terminology. So unlike what you have with eukaryotic initiation factor, et cetera, like we talked about last time. Unfortunately, that's not the case. So just want to finish up with one last common DNA binding motif that you see in eukaryotic cells. It's called the leucine zipper. And you can see the leucine zipper. I talked about this when we very first talked about motifs and structures. You've got 4 cysteine residues, or 2 cysteines and 2 histidines. These bind to zinc and give you a particular alpha helical structure, which sits down into the major groove of DNA. And we'll talk a lot more about these different proteins and regulators that have this particular kind of thing. So we have homeodomains and zinc fingers. We'll talk about some of the other DNA binding motifs on Wednesday. And I'll have your exams by then. I'll have all the scantrons ready.